Okay, so the last screencast on evolution is looking at or reviewing the idea of phylogeny. So macroevolution we talked about yesterday is speciation. So basically we're just looking at how those speciation events occur over a long period of time to create all the diversity of life. And my beloved husband is going to turn down his ninja game.
Ninja game?
Whatever you're playing, dear. I really have to find a way to record these that isn't in my bedroom. Okay, macroevolution is the broad pattern of evolution above the species level. So remember we talked about microevolution as evolution within a particular population, and then macroevolution is evolution that creates different species. So we can look at the emergence of vertebrates, movement of those vertebrates onto land, and these mass extinction events as all sort of major moments in the idea of phylogeny. And the fossil record gives us clues as to that. So we can look at different sorts of fossils. Here we've got fossilized footprints, some fossilized shells, an early human skull, and we can also look at species that are preserved in amber or fossilized in amber. So 512 in the Campbell textbook will give you an idea of how we date rocks and fossils, but we've talked about radiometric dating in different classes. So carbon-14 is constantly, it's a certain percentage of the carbon in the atmosphere. So as you eat things, you take in a relatively constant amount of carbon-14. So you can see here's my lovely little clamshell that's taking in carbon-14 as bicarbonate. You can take carbon-14 in lots of different species, but in this is the bicarbonate because it's in the clamshell. After the clam dies, it's no longer taking in carbon-containing molecules, so that carbon-14 breaks down, and it breaks down at a relatively constant pace. So alive, we're maintaining at a constant level, and then as soon as you die, that carbon-14 begins to break down because it isn't being taken in anymore. So when we look at a fossil, we can look at the amount of carbon-14 in it and get some idea of how old that fossil is based on the half-life of carbon-14. We use that to establish a geologic time scale with a sequence of time periods. So one of the important ones you'll hear talked about a lot in the context of evolution and over different courses is this Cambrian explosion. So that's when we saw by far the sort of largest emergence of most animal phyla. And it's important that we understand what early Earth looked like. So in the primitive atmosphere, we had amino acids floating around and nucleotides floating around. And then those amino acids and nucleotides joined together to create proteins and DNA and RNA. And at the same time, there were sort of early lipids lying around, and those early lipids could then enclose the proteins and the DNA and RNA, forming this kind of protocell. And then those protocells became self-replicating as a result of the DNA and RNA molecules. So we can carry out experiments where we replicate the conditions of early life. Lots of water vapor and carbon dioxide, nitrogen oxide compounds, so various nitrogen oxide compounds, methane, hydrogen, and hydrogen sulfide. And if we simulate that environment by adding lightning or an electrical spark and high UV radiation, we can prove that we can form amino acids and early nucleotides. And that was proven in 1953 by Stanley Miller. And here's his experiment. We've got those early atmospheric compounds. We've got a spark to simulate lightning and a heat source. So this is the water mimicking steam. And we end up finding these organic compounds in the cooled water that comes out of the steam. So then those early small molecules begin to combine and form macromolecules. It isn't necessarily a favorable or a really common event, but it does occur, and the amino acids themselves can start to catalyze some reactions. And so then from there, we get these protocells bound by membrane vesicles, which we'll talk more about. They can form a bilayer, and they actually absorb small pieces of molecules that form a surface on which the RNAs and proteins can adhere, which gives a start to them becoming self-replicating. So we'll watch the TED Talk in class tomorrow. So key events in life's early history, take a look at 514 to 518 of Campbell if you are in the AP course only. This is what I want to talk about. So EVODEVO is a relatively new science. It's evolutionary developmental biology. So what it looks at is how different species evolved and how we developed based more on changes in developmental processes like evolution and then growth and development in early childhood. And what we're starting to understand is that it's not so much a change in gene sequence that causes changes, let's say, between humans and monkeys, but rather a change in when those genes are turned on and off. So most developmental genes are highly conserved, so they stay the same in many species. So we've got this homeobox sequence, for example, associated with genes that regulate the segmentation of the body in fruit flies and animals. So the same genes regulate the development of segmentation in the body. They tell your abdomen to be your abdomen, your head to be your head. They tell the cells in the right places where to divide so that that abdomen becomes the abdomen in the middle and not on the top, and so on and so forth. But if we change those homeobox genes and when they're turned on and off, we can get different signals. So we can change them in different ways. One way we can change them is when they get turned on and off, and that can have huge effects. So we can see that in a couple of different examples. This is a human and a chimp skull. So you can see here is a chimp skull and a human skull at the fetal level, and they look quite similar, strikingly similar. We alter the growth rates of certain parts of the skull. So we change when these genes are turned on and off, and we get a substantially different result. So when we compare a human skull to a chimpanzee skull, you can see that we've got a far less protruding jaw. We've got a far more developed skull, all right? This is a system we call paedomorphosis. So the adult of the human species actually retains the characteristics of a juvenile chimp. And that's because we slow down the growth rate of this jaw and increase the growth rate of the skull, or maintain, actually, I should say, the growth rate of the skull. Whereas in the chimp, that would slow down early on, resulting in the receding jaw of the chimp or the receding forehead of the chimp composed to the protruding forehead of the human. We can also see this change in heterochrony, so the change in rate or timing in the stripe thickness, not the strip thickness, of zebras. So we've got plain zebras and greavy zebras. Think about if you took a balloon and you inflated it. So you drew the stripes on the balloon before you inflated it. The more you blew up the balloon, the thicker the stripes would get. But if you didn't draw the stripes until the balloon was already bigger, they wouldn't have time to get as thick as they would if they'd been turned on right from the very beginning. So if we turn the gene on later in this greavy zebra, you can see we get much thinner stripes compared to the plain zebra, where that striping gene, the melanin-producing gene, is turned on much earlier in development. We can also change the homeobox genes to change the spatial pattern of organisms. So if we change the genes, we can change them so they control placement and spatial organization differently. So these Hox genes are involved in controlling where body parts go. In crustaceans, the UBX and SCR Hox genes convert a swimming leg into a feeding claw. And in snakes, these Hox genes, HOXC6 and HOXC8, are what's responsible for suppressing limb formation. So you can actually see underdeveloped skeletal limbs in snakes. They just never form out, and that's because these two Hox genes are turned on to suppress that formation. And what can happen is that we get gene duplication events occurring. You can't have changes in a developmental gene really and have a successful offspring unless you have multiple copies of that gene. So if you only have one copy, you can't afford to screw it up. If you screw it up, the embryo is going to be inviable 90% of the time, or certainly it won't live very long. If you have multiple copies of the gene as a result of a duplication event, then you can screw around with one or the other of the genes and you won't experience a problem. So we can have gene duplication changes and other regulatory gene changes creating novel forms. So going back to that UBX example, modern insects developed from early crustaceans. In insects, UBX suppresses limb development in the abdomen. What that means is that only the thorax, the middle of the bug, grows limbs. So if you look at a bug, bugs have a head, a thorax, and then an abdomen at the butt. And if you look at where their limbs are only coming out of the middle, the thorax. If you remove UBX from an insect, so in this case, the fruit fly, it grows legs all over, including on its abdomen. If you activate UBX in the thorax, where it's not normally expressed, the fruit fly won't develop appendages. It won't develop legs or wings. So you can take that UBX gene out of a crustacean, so out of something like a lobster, stick it in a fruit fly, and you can see that it'll suppress some fruit fly limb development, but not all. And that's because the sequence of the UBX gene in the crustacean has changed over time. It's not identical to the UBX that's in the fruit fly. But if we do mutate that crustacean gene a little bit to make it more closely resemble the insect UBX, then we'll see the suppression increase again. So this kind of study allows us to, again, estimate that phylogeny more accurately. It gives us some sense of how closely related things are. So here we could look at these UBX genes and the more limb suppression we see in the fruit fly, that tells us it's more closely related. So if we took a UBX gene from a housefly, we'd see substantially more suppression than we would from a crustacean. We might be able to take one from a spider, who are more closely related to crustaceans than insects, and again, we'd see maybe that 15%, maybe more, maybe less. We can also have changes in gene regulation, and the idea, we can also talk about the idea of evolutionary novelties. So evolution is influenced by a variety of different factors, but it isn't goal-oriented. It doesn't proceed in a direction per se, and the human eye is a good example of that. So typically when we talk about phylogeny, we're talking about the evolutionary history of a species from its common ancestors. And the discipline devoted to phylogeny and classifying those organizations and determining those relationships is called systematics. And it uses fossil data, molecular data, and genetic data to infer those relationships. So it's a little bit different than taxonomy, which is simply identify it, name it, classify it. And we remember Carlos Linnaeus, he's the one who developed the binomial nomenclature system. So we have that hierarchical classification to review. So we've got species, genus, family, order, class, phylum, kingdom, and then now kingdoms, located above kingdoms in our understanding is domains. So we have 3 domains of life: archaea, bacteria, and eukarya. And we use a phylogenetic tree to represent the phylogeny, and the classification should match. So a lot of organisms have actually been reclassified, put into a new genus or a new family, based on our increasing understanding of phylogeny and genetics. So I talked about yesterday how in some cases things that we had morphologically classed as species we realized weren't actually their own species. So if we look here, we can see the phylogeny or the relatedness between a group of common mammals. They're all part of the order Carnivora. If you go back farther, they would be Mammalia and then Animalia. So the order is Carnivora, and then you can see we split here. So dogs and cats, let's call them, split after carnivores. So the two groups of carnivores can be subdivided into the dog half and the cat half. So really you can be a cat person or a dog person. It starts fairly early. So here we've got the family Felidae, and this is going to be all of the sort of cat-related things. And then this particular cat belongs to the subgroup Panthera and Panthera pardus, or the leopard. Okay? The dog group then breaks into a further sub-classification. So you have what we would call the sort of dogs and dog-related, and you also have the sort of weird things that are sort of mammals but not quite dog-like or cat-like that we can see now are more closely related to dogs than they are to cats. And these examples are the striped skunk and the European otter. So there are obviously more species within the genus Mephitis, which would be the skunk genus. There are obviously more otters within the Lutra genus, which would be all the otters. So we've got sea otters and this, that, and the other thing. And then our Canidae family consists primarily of the genus Canis. And then we've got different organisms within there. So Canis familiaris, which is the domestic dog. We've got Canis lupus, which is the wolf. You've also got Canis latrans, which is the coyote in here too. And sometimes things in the same genus can breed with each other. So we can have wolves and dogs breeding. We can have wolves and coyotes breeding, so on. The way we read a phylogenetic tree to review, the common ancestor is here at the branch point. So at this base of this tree is the ancestral lineage, and 1 is the last common ancestor of all of these taxa, A to F. The branch point here, or node, is the last common ancestor for A to C. So we call this spot where they come off a node. So 4 again is another node. 3 is a polytomy. So if you take a look at taxon DEF, we have 3 things all coming off the same branch point. And what that tells us when we see 3 things coming off at the same time, as opposed to splitting later on like this, is that we really aren't exactly sure what happened here with D, E, and F yet. So we're not really clear on the phylogenetic relationship. We don't know whether F came off of E, whether E came off of D, whether D came off of F, what have you. The other thing I just want to point out is if 2 things branch off from the same common ancestor at the end, we can call them sister taxa. So B and C, for example, are sister taxa. And just the only term that we didn't discuss there is the basal taxon. So the root of the tree, I'm just going to go back down here to ancestral lineage. That's the basal taxon. And we can describe our tree as rooted if everything on it comes from the same common ancestor. So to create a phylogenetic tree, what we're looking at are homologies, and we've talked about homologies. So we're looking for homologous features that allow us to put things in the same spot. But we've got to be careful. We're looking at homologous structures as opposed to analogous structures. So here we've got the Australian marsupial mole and the North American mole, and they have analogous structures, but they aren't that closely related. The marsupial mole is going to be more related to the other marsupials in Australia than they are to that American mole. And so this method of inferring the relationship from homologous characteristics is called cladistics, and we group species into clades, which are the common ancestor and then their descendants. But the goal at the end is to have a monophyletic group. So a monophyletic group here, we've got a common ancestor and then all of their descendants. A polyphyletic group, you can see here we've got E, F, and G. So it ends up being two different common ancestors. And a paraphyletic group includes the common ancestor and some, but not all, of that ancestor's descendants. So shared characteristics are, again, what we use to classify. So we're going to look at homologous characteristics. We're going to look at currently shared characteristics. So shared ancestral characters originate in the common ancestor, so backbones, invertebrates. Everything in the vertebrate clade is going to have a backbone because that originated from some common ancestor. Whereas a shared or derived characteristic is something that are unique to that particular clade. So for example, marsupials have pouch development. Mammals have hair. They have amniotic offspring, things like that. So we can construct a cladogram similarly to how we would construct the phylogenetic tree. We choose an organism to be our in-group. So we pick the organism that we're trying to study, and then anything that isn't in your in-group is in an out-group and has to diverge somewhere before your in-group. And so we do that by kind of saying, okay, our leopard is my in-group. So we can... sorry, my parents are calling apparently, and I'm going to ignore this phone call. So the vertebral column will separate out first the lancet. So the lancet becomes the first outgroup, and then from there, now everything else has a vertebral column. And then we can say, okay, everything here has jaws, so we can separate out the lamprey. And then we look at what's left. So we've got the tuna, the salamander, the turtle, and the leopard. So we then separate it out by, we have legs. Okay, the tuna doesn't meet that, so the tuna now becomes the outgroup. So we're left with the salamander, the turtle, and the leopard. So then we move to, has an amniotic egg. So now the salamander becomes an outgroup. They don't have an amnion in their egg. We're left with turtles and leopards. Well, the turtle doesn't have hair, the leopard does. So that's our final in-group. And they've represented that here by just adding ones. So you get a 1 if you meet that criteria, and you get a 0 if you don't. And this is called a character table. So here's another example of a cladistic group. And this shows you how the character would translate. So this is 1. This would be a 1 and a 1. Anything where there's no colored line would be the 0 on the character table. So this shows you the relatedness between the eastern box turtle and the duck-billed platypus, red kangaroo, North American beaver. So maximum parsimony and maximum likelihood are what allows us to sort through the DNA sequence. So when we're trying to do classification, sometimes there's just so much information we have to kind of make a hypothesis based on that information. And we usually make the hypothesis based on 2 principles. Maximum parsimony basically is sort of like Occam's razor. So if you know what Occam's razor is, basically it's the simplest explanation that's consistent with the facts, is the most likely to be true. Not truer, I really have to edit these PowerPoints. Maximum likelihood is that given the probability of mutations in the tree, we should construct a tree that represents the most likely series of events. So we don't want to construct a tree that assumes that humans evolved from birds and once had wings and lost them. We want to assume that somewhere after the vertebrate, humans and birds diverged, if that makes sense. We can use molecular clocks as well. because certain genes are known to evolve in a relatively constant rate from time to time. So we can look at changes in these genes from species to species, and the number of mutations will give you an idea of how long it's been since those species diverged. They have some limitations, though. So that is phylogeny.